Hello and welcome to the WeActuary Online Academy. My name is Chris and I'll be your tutor for today. And today we'll be looking at the Cambridge AS Biology paper, course code 9700, and we'll be doing the multiple choice paper 1-1 of winter 2019. Okay, this video is part 1 of 8 and we look at questions 1 to 5 in this tutorial. Question 1. Which cell structure contains cytoplasm? Okay, so before you answer this question, you might have thought about the endosymbiont theory that the mitochondria and chloroplast were once prokaryotic cells that became a part of eukaryotic cells later on. So you might think either A or B could be the answer here. But this question is referring to the cell structure that contains the cytoplasm. It's really referring to the cytoplasm of the cell itself, okay, rather than the internal structures of each organelle. So here we have the chloroplast. Now inside the chloroplast they have the stoma. Inside the mitochondria you have the matrix. Okay. Now although there's both liquids inside these, they're more gel-like and a bit more viscous than the actual cytoplasm of the cell itself. Okay, but remember it's a double membraned bound organelle and it's separated from the main cytoplasm of the cell. All right. However, the plasmodesmata, if we remember, this effectively creates pores in between the cell wall of adjoining plant cells. This allows the cytoplasm of the plant cells to move freely with one another without any kind of resistance at all. So option C is the answer here. Okay, option C, the plasmodesmata does contain the cytoplasm of the cell. The smooth endoplasmic reticulum, well, they have the cisternae, but inside there is just fluid substances containing soluble salts and other kind of nutrients in there as well. Okay, so option D is incorrect also. Question 2. Some cell structures are listed in a particular order below. We have the nucleus, ribosome, Golgi body, and vesicle. What determines the order in which these cell structures are listed? Okay, well, we have our options here. Let's go through some of the easy ones we can eliminate. Okay, so let's look at D. Size smallest to largest. Well, no, that's incorrect because the nucleus is the largest organelle. From largest to smallest, well, that's also incorrect because number 2, ribosome, is the smallest structure. So let's look at what structure number 3 is. Remember, the Golgi apparatus is used in the modification of proteins, okay, of proteins, okay. So if we look at our two options here, synthesizing of a lipid or synthesizing of an antibody, well, an antibody is a protein. So the Golgi apparatus is most likely involved in the synthesizing of this antibody, and then it will create a Golgi vesicle, which would then fuse with the membrane and release it into the extracellular environment. All right. Now, for the synthesizing of a lipid, this involves a smooth endoplasmic reticulum, which is not an option here. So the only option available is option B. Question 3. The drawing has been made from a section showing part of an alveolus and a red blood cell in a capillary. The magnification of the drawing is times 5,000. All right. Now the question is asking you, what is the minimum distance oxygen must diffuse from the air in an alveolus into the red blood cell? Now the minimum distance would be here, this location right here. Okay. So if we remember our formula, if you remember, you've got your magnification triangle. Okay, we've been given what M is already, so we know what M is, and we can work out I based on the value of this measurement here. So if you got a ruler out and measured it, this would be a total of 5 millimeters. Okay, and remember what you should do then is if we're working out actual size, it would be I divided by M, but what you should do first is convert 5 millimeters into micrometers, which is by timesing 1,000, which gives you a value of 5,000, and the magnification value is times 5,000, and therefore if you do the math, it works out as 1.0 micrometers, which is option D. Okay, so you really need to do your maths here and make sure you're not confused in the unit between millimeters to micrometers. Okay. Question 4. 4 students were asked to match the function with appearance of some cell structures in an animal cell. The functions were listed by a number. Number 1, mRNA passes through to the ribosome. 2, synthesizing of polypeptides. 3, packaging of hydrolytic enzymes that will remain in the cell. The appearances were listed by letter. V, membranes which surround an enclosed inner cavity. W, non-membrane bound spherical structures. X, a double membrane interspersed with pores. Y, non-membrane bound cylindrical structures. Z, membrane bound sacs arranged as a flattened stack. Okay. Which student correctly matched the numbered functions with the appearance of the cell structure? Okay, let's look at what each of these numbers represents then. So when the mRNA passes through the pores, okay, they're really talking about the nuclear pores here. The nuclear pores inside the nuclear membrane or the nuclear envelope. Okay, so if we look at the options here, the only option that refers to pores and a double membrane is X. So X is correctly describing the nuclear pores in the envelope. So I can eliminate B and A from my choices here. Now, before I look at question 2, the idea here is you've got a choice between W and Z. Okay. Now, 2, synthesizing of polypeptides takes place in the ribosomes. Okay. Now, if you're unsure, you could use a process of elimination here. Whereas, so if we look at, let's say, option D, you have Z, membrane-bound sacs arranged as a flattened stack. Well, this is really referring to the smooth endoplasmic reticulum. In this case, you could have maybe taken a guess. It could have referred to the Golgi at that point as well. But even still, you don't really know. However, ribosomes do not have any membrane. They're described as small spherical organelles. They're very small. I mean, if you look at a picture under very high magnification, the subunits that it makes up don't exactly look spherical, but they are correctly described as spherical organelles. Okay, so that means 2 refers to W. Okay, and therefore option C is the correct answer, eliminating D. All right. Question 5. The antibiotic chloramphenicol inhibits protein synthesis in the mitochondria and in some prokaryotics. Chloramphenicol does not inhibit protein synthesis in cytoplasm of eukaryotic cells. What would be the effect of a person being treated with chloramphenicol? Okay, well, this question you might get lost in the words. So it clearly says in the mitochondria, okay, and in some prokaryotes, although this bit of the statement is irrelevant because it's really talking about inside of a person, not prokaryotics. So protein synthesis in the ribosome still occurs as normal. However, the mitochondria does carry out protein synthesis, and it makes its enzymes as well. Just in order to make ATP, it has to make the enzyme ATP synthase. So if it's not able to build its proteins, then it will not make this enzyme ATP synthase. And as a result, there'd be less energy available. Now, the transcription of nuclear DNA is an active process. It does require ATP. So if there's less ATP, then there's going to be less transcription of the DNA itself. Now, this leaves us with option A. Okay. The fact here that we could have eliminated option D and C by the fact of understanding that mitochondrial protein synthesis is important for ATP production. You stop it, less going on. Now, of course, if there's less ATP, there's going to be a negative effect on all active processes. This is an active process and does need energy for that. Okay, so we eliminated option B. Thank you for watching our video today. Please register on our website for a free demo, and I look forward to seeing you in the next video.